Welcome back, students. Welcome to Priya's Life Science Academy. I hope you are doing well. Today I am back with another very important chapter of genetics, which is gene mapping or linkage mapping. Pay attention and listen carefully. This chapter will be beneficial for those who are preparing for CSIR NET Life Sciences, ICMR, GATE Life Science, NEET, and for those who are in bachelor's degree or in master's degree. So let's start. So before starting, I want you to understand a very important part, which is crossing over. To understand what gene mapping actually is, you need to understand the concept what is crossing over. You know that during meiosis, particularly during gamete formation, there is a phenomenon known as crossing over which occurs. Okay, in meiosis it occurs, and it occurs between two homologous chromosomes. You know what are homologous chromosomes? I hope you understood it very clearly from my previous lectures, that we have two homologous chromosomes for each gene. One part is coming from mother and another chromosome part is coming from mother. So between two homologous chromosomes, crossing over occurs during prophase 1 of meiosis. Okay. So what is crossing over? Crossing over occurs during prophase 1 of meiosis, specifically in the Pachytene stage. During this stage, homologous chromosomes pair up and exchange genetic material at specific points known as chiasmata. You can see here in the picture that homologous chromosomes, they are pairing up with each other, and there is a crossing over. Crossing over means this blue chromosomes part is coming into the red chromosome, and this red chromosomes part is coming into the blue chromosome part. So this is known as recombined genetic information or recombined genetic material, where the chromosome segments are exchanging their parts with each other during meiosis 1 of prophase 1 in the Pachytene stage, which is very much important for bringing genetic variation in organisms. Okay. So, you know, during gamete formation this occurs. That is why there can be so many combination of gametes. Okay. So, this process increases genetic diversity, I told you. Now, the question is that where does crossing over happen? Between homologous chromosome. You should remember, between homologous chromosome crossing over happens. You know where crossing over occurs? And I told you that crossing over always occurs between two homologous chromosomes. One is inherited from father and one from mother. Number 2, it does not occur between sister chromatids of the same chromosome because they are identical and do not contribute to genetic variation. These two are sister chromatids. Okay, the crossing over doesn't occur between two sister chromatids. These two are sister chromatids. Crossing over doesn't occur between two sister chromatids. Crossing over occurs between two non-sister chromatids. Okay, now where they occur mostly, crossing over occurs between genes of the same chromosome if they are far enough apart. So I told you crossing over occurs between two non-sister chromatids, but when it occurs between two genes on the same chromosome, the position of the two genes on the same chromosome must be specific. There must be a long distance between the two genes. If crossing over occurs between two genes on the same chromosome, there must be a long distance between the two genes. Okay, so crossing over occurs between genes on the same chromosome if they are far enough apart. Okay, now distant genes, if two genes are far apart on the same chromosome, crossing over is more likely to occur between them because there is a greater physical space for recombination to take place. So in order to occur recombination, you need to have a large distance between two genes. Okay, so between two genes there must be a large space. So between two genes there must be a large space for recombination to happen. And what about close genes? If two genes are very close to each other, crossing over between them is less likely because of limited space. Such genes are tightly linked and often inherited together. Now comes about linked genes. Okay, so there are 2 points which you need to consider. One is the crossing over between two distantly spaced genes, and one is linked genes. Like when the genes are very closely spaced, they tend to inherit together from one generation to the next generation, okay? Or during gamete formation, if the genes are very closely spaced in a chromosome, these two genes will be transmitted or inherited together in the gamete. There will be no crossing over. In order to have crossing over, you need to have a space, a physical space between two genes. So from this part, what you understood, that crossing over always happens between two homologous chromosomes, between two non-sister chromatids, which is the main thing to bring genetic diversity. And recombination also happens. Recombination happens between two genes when they are distantly located, but not between the genes which are closely spaced. So in that case, we have another term which is linkage. Linkage is nothing but when two genes tend to inherit together. That is known as linkage or linked genes. Okay, so you can see here that distant genes on the same chromosome are more likely to undergo crossing over. In fact, if the genes are far enough apart at opposite ends of the chromosome, the likelihood of crossing over between them approaches 50%, okay, making them appear to assort independently as if they were on the separate chromosome, okay? So if two genes are very distantly located, then in between them, crossing over, the chance of crossing over approaches to 50%, and they can assort independently, okay, as if they were on the separate chromosome. In contrast, genes that are closer together on the same chromosome are less likely to recombine because the physical distance between them reduces the chances of crossover event. So if the two genes are very, very closely spaced, there is no point of crossing over. There is no point of crossing over. In order to happen crossing over event, you need to have a physical space between them. So when the genes are very closely spaced, they are less likely to recombine because the physical distance is very less, and that itself reduces the chance of crossing over event. Okay, so the main takeaway message from this slide is that homologous chromosomes are involved in crossing over. Crossing over occurs between non-sister chromatids of homologous chromosomes during meiosis 1. Point to be noted. Genes on the same chromosome. If it happens genes on the same chromosome, then what will be the condition for closed genes? Less likely to cross over because they are tightly linked. For the closed genes will not recombine with each other because they are very, very closely spaced. And for distant genes, more likely to cross over because they are loosely linked or behave like independent genes. So if the genes are on the same chromosome, what's the condition of the crossing over? If the genes are on the same chromosome, you have to notice, if the genes are on the same chromosome, if they are very closely spaced, then there will be no crossing over. But if the genes are distantly spaced on the same chromosome, then there will be the chance of crossing over. Okay, now independent assortment and crossing over. Okay, so these two are very, very important drivers of creating diversity in organisms. One is crossing over during meiosis 1, and after that during gamete formation, that's independent assortment. Crossing over creates recombination, which breaks linkage and increases genetic variation. If genes are far apart on the same chromosome, they behave as if they assort independently because recombination occurs frequently. Okay, so these two are very important. One is crossing over and another is independent assortment, that after crossing over, the recombined chromosomes will assort independently into different gametes, giving rise to different combination of gametes. I'll show this to you later. So to understand gene mapping, first you needed to understand this, that what is linked gene, what is crossing over, where does crossing over occurs, and everything. I hope you understood. Now coming to gene mapping, so there are three types of mapping. actually. Genetic mapping, physical mapping, and comparative mapping. Today we will deal with the genetic mapping, that is a linear description of DNA markers or genes on a given chromosome with closely spaced markers being inherited together more often. So when what's the position of the genes and what is the distance between two genes when it is calculated by calculation of recombination frequency or crossing over, that's known as genetic mapping. But physical mapping is different from genetic mapping, that is which denotes the physical location on the chromosome, relating more towards exact positioning of the gene elements, okay. And comparative mapping is gene sequencing, okay, databases, DNA chips, which are very, very important for forensic studies. So linkage mapping between versus physical map, like it follows, the linkage map follows, it's a chromosome that shows the position of its known genes relative to each other in terms of recombination frequency. I told you before that in terms of recombination frequency, what's the position of the genes, what's the distance between two genes. And physical mapping is that it's a specific physical distance along each chromosome in physical maps. Linkage map is depending on recombination frequency and physical map is depending on actual physical distance between two genes. Now this is the genetic linkage analysis. Here you can see that in G1 interphase, that's these two are homologous chromosomes, and after replication these two are sister chromatids. After replication these two are sister chromatids, and between these two homologous chromosomes recombination happens during meiosis 1, prophase 1. The strand exchanges happen. You can see here. So the red part comes here and the green part goes there. Now what happens? This is the metaphase 1 phase. During metaphase 1 phase, there is an independent assortment. Gametes assort independently. This is the total green gamete goes here. This is the total red gamete comes here. This is the green and red gamete, this comes here, and this is the red and green gamete comes here. So these two are parental combination, and these two are recombinant. Okay, so during meiosis 1, homologous chromosomes exchanges their strand and the recombined gametes are produced. Okay, here by another example, here you can see in the parental chromosome, there in these two are homologous part. Here is capital A, capital B, here is small a, small b, but when they are recombined, you can see here, so this is a parental type and this is recombinant types. Capital A comes with small b and small a comes with capital B. So you can see here this type of recombination happens. There can be two type of mapping: 2-point test cross, 2-point cross, or 3-point cross. 2-point cross is distance between two genes, and 3-point test cross is distance between 3 genes. Okay, and now who gave us the idea of this? T.H. Morgan. Okay, T.H. Morgan gave us this idea of recombination frequency crossing over. So crossover frequency is proportional to distance between loci. It's a very, 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 very important thing that you know that crossover frequency or recombination frequency is equal to the distance between two genes. If I say the distance between gene A and B is 20 centimorgan, that means the recombination frequency between gene A and B is 20%. Or vice versa. If I say that the recombination frequency between gene A and B is 20%, then their distance is 20 centimorgan. So recombination frequency is equal to distance. RF for recombination frequency and distance they are equal. So the relationship is very easy. 1% recombination frequency is equal to 1 map unit or 1 centimorgan. Cm for centimorgan, okay, and mu for map unit. So 1% recombination frequency is equal to 1 map unit or 1 centimorgan. The unit of the distance is denoted in this unit, centimorgan unit, okay. Now we will look into the recombination or crossing over more closely. Here you can see this is the parental generation. This is one parent, this is another parent. When they are crossed, we are getting this offspring. So what type of gamete will come from here? From this capital A, capital B will only come, and from this small a, small b will only come, and when they recombine, they give us this type of offspring, capital A, small a, capital B, small b. Now the next part is the test cross. What is test cross? That with the F1 offspring with the one of the parent who is recessive, okay, homologous recessive. So we are doing the test cross. Now this is our cross. Now the question is, the number of offspring is 1000 from this cross. Parental combination is 800. Total number of offspring is 1000. From them parental is 800 and recombinant is 200. Now we will understand that how this recombination actually works. Okay, so I'm going to another page. So the parent was capital A, small a, capital B, small b with small a, small a, small b, small b. This is the test cross. So let's write their chromosomes. These are the gametes for this parent, and these are the gametes for this parent. Now you can ask me the question that why, ma'am, you wrote capital A and capital B in the same chromosome? You could also write capital A with small b, but why did you write capital A with capital B? Because I know the parent. You know the parent was capital A, capital A, capital B with small a, small a, small b, small b. So the offspring was, that's why capital A, small a, capital B, small b. So this capital A and capital B came from the same parent, so they must be on the same chromosome. That is why I wrote capital A and capital B on the same chromosome. Now these are the gametes of these two parents. Now let's do. So now there will be crossing over. Okay, now you will get different types of gametes from here. These are the gamete combination of the parents when there is no crossing over. Now if there is crossing over, how many types of gametes you can get? So from here only, simple, capital A, capital B, but from here capital A goes here with small b because crossing over occurred. Now then small a didn't go straight, it goes here with capital B, and it is normal, small a, small b. So you know, here you can notice that before crossing over these were the type of gametes, but after crossing over there are different types of gametes. These two are parental combination of gametes and these two are recombinant gametes. Okay, and from these how many types of gametes you can get? Only one combination, small a, small b, because even if here is recombination occurs, crossing over occurs, small a will go with small b, small a will go with small b. So here you can notice that after crossing over, how many types of gametes can be produced. Now we will combine these two types of gametes from each parent. So how many types of offspring we will get? So from here capital A, capital B, from here small a, small b, from here capital A, small b from here, small b from here, small a capital B from here, small b from here, small a capital B from here, small a small b. So here you can notice by combining these two types of gametes from each parent, this is parent 1's gamete, this is parent 2's gamete. So when we combine them, we could get lots of different types of offspring. This offspring is of parental combination, this offspring also, because capital A capital B small a small b, these two are parental combination. But look at these two types of gametes, look at these two types of offspring. These two types of offspring are recombinant offspring. So you understood that by crossing over there will be different types of gametes, and when different types of gametes are combined, they will produce different types of offspring. Okay. Now there can be another question in your mind that why ma'am you wrote this in double dose? Because I wrote this in double dose because the chromosome will duplicate during replication. Okay, these two are homologous chromosomes of each other, and these two are non-sister chromatids. Only non-sister chromatid participate in crossing over. Okay, but these two extreme chromosomes, they do not participate in crossing over. And these two are homologous chromosomes of each other. I duplicated them because you know the chromosomes are duplicated after replication. Now see here. Now I'll show you another example. Here it's a 3-point test cross. I showed you the cross between two genes. Here I will show you the cross between three genes A, B and C. Okay, so A, B, C, they are also duplicated because after replication they are duplicated. Then small a, small b, small c. Okay, so these two are homologous chromosomes, and when crossing over occurs, you can see here there are three types of crossing over between the three genes. One between A and B, okay, one between B and C, and one between A, B, B, C both, which is known as double crossing over. Okay, so what is double crossing over? When crossing over occurs between A, B and B, C both. So from it we will get different types of offspring. Okay, so by this slide what you could take away that when there are three genes, there is a possibility of a double crossover event as well. But you know, double crossover event is not so evident always. It's a rare case. Okay, whenever there is a single crossover, it mitigates or suppresses the chance of another crossing over event, which is also known as interference. I will come to this later. Now here you can see this is one parent. So from this parent, when there is no crossover, this is the parental gametes which we can get. When there is a crossing over between A and B, so what type of gametes we can get? Capital A will not go with capital B, it will go down with small b and small c. So capital A with small b and small c. And next, small a will not go with small b, small c. Small a will go up with capital B and capital C. Look, small a with capital B and capital C. So there is a single crossing over event between A and B. So we could get one recombinant type of gamete. Again, if there is a crossing over between B and C, this was between A and B. Now this crossing over event is between B and C. Now A, B will not go with capital C. A, B will go down with small c. Look, A, B with small c. Then small a, small b will not go with small c. It will go up with capital C. So small a, small b with capital C. This is another crossing over event, and from it also recombinant type of gamete are produced. Now, when crossing over occurs between A, B and B, C both, what will happen? So capital A will go down first with small b, then will go up with capital C. So capital A with small b, small c. Then small a with capital B and it will go down with small c. So capital A with small a with capital B and small c. So this is the double crossover event. This is another recombinant type. So whenever there is a 3-point cross, you need to consider that there will be four types of offspring or 4 types of gametes. Number 1, parental combination when there is no crossover. Number 2, single crossover between the first 2 genes. Number 3, single crossover event between the last 2 genes. And number 4, the double crossover event between the 3 genes. Okay, now here is a question where it is given geneticist is studying a 3-point test cross where he got this following gametes and their observed count. So these are the gametes, these are the genotype of the gametes, and these are the numbers observed for each gamete. So before starting, I want you to know these points very clearly, that the parental combination or non-crossover combinations are highest in number. The parental combination is always higher than the recombined combination because the chance of getting the recombinant types is lower than getting the parental types. Okay, so the parental combination will be highest in number. So when you get the table, there will be the numbers of the gametes. Okay, and these are the genotype of the gametes. Okay, first you have to take out the gametes which are highest in number and denote it as a parental gamete. From here what you can see, 340 and 360, these 2 gametes are highest in number. So from this you can understand that these 2 are non-crossover gametes or parental gametes. So let's write then. Capital A, capital B, capital C, small a, small b, small c. So capital A, capital B, capital C, this is 340, and small a, small b, small c, this is 360. Point to be noted that these are gametes. Gametes are always singular in number, that is n. But when I am writing these 2 gametes in this fashion, this is known as an offspring. Children often confuse with this, that this is one gamete, this is one gamete. When n and n they come together, they become 2n, and that becomes an offspring. Okay, you are given the number of gametes produced for each genotype. These are the gametes. So first you identified the 2 gametes of the 2 parental gametes. Then you have to identify the double crossover gametes. And here you also have to notice one thing, that the percentage of double crossover will be the least in number. Okay, so the parent combination will be highest in number, and the DCO or double crossover will be least in number. Why? I told you that one single crossover always interferes with the occurrence of another crossing over. That is why there will be a very less chance when there will be a crossing over between A and B and between B and C also. Okay, so the double crossover event will be least in number, so their percentage will also be lowest. And in between the parental combination and DCO combination, there will be the percentage of single crossover event between A and B and the single crossover event between B and C. So in between them, there will be the single crossover event between A and B and single crossover event between B and C. So here you could see that our NCO is this, and which is the lowest number? 15 and 10. So this is the NCO class, this is the DCO class. NCO means non-crossover, DCO means double crossover. Now what's the question given? Which gene is in the middle? That is they are asking about the gene order. Calculate recombination frequencies. What are the recombination frequency for each interval? Recombination, and I also told you recombination frequency is equal to the gene distance, the map distance, the map distances. They are asking, construct a genetic map showing the distances between the genes and interference calculation. And what is the interference? So first of all, what you have to know, that you have to know the gene order. that which gene is in the middle. You need to know that. So in order to do that, you need to arrange the NCO in that order that you can get the DCO very easily. So in order to understand that what is the middle gene or what is the gene order, you need to write the NCO in a specific order. If I write the NCO as it is, like capital AVC, small avc, and if I do the double crossover, like capital A, small b, capital C, capital A, small b, capital C, and at down small a, capital B, small c, small a, capital B, small c, and you have to match it with the given DCO configuration. So here you can see capital A, small b, capital C matched, small a, capital B, small c matched. Oh, it is matched. So if the given NCO's configuration matches with the DCO given after you do this cross DCO, then your gene order is like that, like A, B, C. So your gene order will be like the NCO's order, like A, B, and C. So what is the gene order and what is the middle gene? The middle gene is B. If the gene order doesn't match with the given NCO, then you have to change it like that, like C, A, B, C, A, B. Then you have to cross like C, A, capital B, then C, A, capital B, small b, then you have to match it with the given DCO if the DCO matches with it or not. So here the question is very clear. So your DCO, your gene order is A, B, C. Okay. Now what is the recombination frequencies for each gene, or what is the map distance? So in order to take out recombination frequency, there is a formula. Recombination frequency is equal to number of recombinant offsprings divided by total number of offspring into 100%. Okay, so this is the formula. So first you have to take out the total number. The total number is 1125. Sometimes it is given, but sometimes you have to take it out by your own. Okay, so first I have to take out the gene distance between A and B, or recombination frequency between A and B. So recombination frequency between A and B is equal to number of recombinant offspring. So you have to know which are the gametes that denotes the single crossover events between A and B. So let's write the NCO class. Now do the single crossover event now. So if I do single crossover event, capital A comes with small b, small c, capital A comes with small b, small c, and in the down, small a comes with capital B, capital C, small a comes with capital B, capital C. So find here where it is the capital A, small b, small c. Here capital A, small b, small c, and small a, capital B, capital C, small a, capital B, capital C. So this is the single crossover gametes between A and B. So their numbers must be written. So number of recombinant offspring. So number of recombinant offspring, the formula. So recombination frequency between A and B is 120 plus 130 plus the DCO, the number of DCO, because you know in the DCO also there is the presence of A and B. What is DCO? DCO is the double crossover between A and B and B and C. But in the recombination frequency between A and B, there is a percentage of DCO, because DCO comprises of A and B and B and C. So the SCO1, it is A and B, the recombination frequency or map distance between A and B is also known as SCO1, that is the single crossover one. So 120 plus 130 plus 15 plus 10, and the total number 1125 into 100% which gives you 24.44%, or it is called as 24.44 centimorgan. So the distance between A and B is 24.4. Okay, so this is the distance between A and B. Now if I want to take out the distance between B and C, then so this is the NCO class done. This is the single crossover event, this is the double crossover event. So this is left only, this is the single crossover event 2. Again we want to assure it with the NCO class. So now this will be the crossover between B and C. So the gametes will be capital A, capital B, small c, capital A, capital B, small c, small a, small b, capital C, small a, small b, capital C. So here you can see capital A, capital B, small c, small a, small b, capital C. So it also matches. So here the recombination frequency between B and C, or the also known as the map distance between B and C is equal to 80 plus 70 plus the DCO. I told you that DCO is present in SCO1 and SCO2 both, because DCO is comprises of crossing over between A and B and also between B and C. So total number of recombinants divided by the total number of offspring 1125 into 100%. So it gives you 15.6%, or 15.6 centimorgan. So between A and B, B and C, so between A and B 24.4 centimorgan, between B and C 15.6 centimorgan. So these are the distances between A and B and between B and C. So I hope you understood this type of problems. Here is another problem, same type of problem. Here again you have to find out first the NCO class. Here you can see 82 is the large number, 70 is the large number. So write it here, N U small t, then small n, small u, capital T. So 82 and 70. So this is the NCO class, and the smallest is 2 and 4. So capital N, capital U, capital T, and small n, small u, small t. So 2 and 4. Now you have to understand that what will be the NCO class gene order to get this DCO. First, if I do with the given NCO's configuration, that is NUT, then what will I get? NUT, N small u, small t, small n, capital U, small t. Do I get this DCO's configuration? No. Now if I change the position of these, so N in this position, T in its place, U in this place, so you have to change the middle position every time in order to get the DCO. So N T, now T in the middle. So at first we did U in the middle, now T in the middle. Now let's see if I get here N T U, then N T U. Now here you can see the position is matching. So at the upper position, capital N, capital T, capital U, capital N, capital T, capital U, and in the lower position, small n, small t, small u, small n, small t, small u. It doesn't matter whether here U is in the middle or either here capital T is in the middle. You need to match all the things here. Okay, like capital N, U, T is in the upper position. Here also capital N, T, and U in the upper position. You need to match these. You don't need to match the order, but you need to match that every letter is in the upper position and every letter is in the lower position. In the lower position also small n, small u, small t, small n, small u, small t. Now you have to understand which letter you put in the middle of NCO to get this DCO, you put T in the middle of this NCO class to get this DCO. So your T is the middle gene here. So NTU, or it can be UTN, but T is in the middle gene. Okay, now in order to find the distances between them, what you need to do? NTU, because T is in the middle gene. Now we want to take out the distance between N and T, which is also known as the single crossover event. So let's find the NT, let's find the distances, let's find the gamete. So if we do the single crossover, then NTU in small t small u, then small n capital T capital U. So we know this was the NCO class, this was the DCO class. Now we have to find single crossover event and one single crossover event 2. So capital N small t small u, capital N small t small u. So this one we got 17 plus small n capital T capital U, small n capital T capital U. This is 13. So the distance between N and T will be number of recombinants into 100 divided by total number of offspring. So number of recombinants, so the distance between N and T we found out. This is the NCO class. By writing the NCO class, we will try to find out the gametes of single crossover. So we did this crossover event and we understood that which gametes are of single crossover, and we know that in the single crossover event we have to write also the double crossover or the DCO class, because in DCO single crossover event 1 and single crossover event 2 both are present. So the DCO class is 2 and 4, that is 6 into 100 divided by the total number of offspring is 248. So what comes? 14.5 centimorgan. So the distance between N and T is 14.5 centimorgan. Now the distance between T and U, that is this is the single crossover event 1, this is the single crossover event 2. Now what will be the distance? Now let's do it for T and U. This is the NCO class. We have to write all the time and we have to find the single crossover event 1, single crossover event 2. Now let's find single crossover event 2. We need not to find here because these two only are left, but let's match them whether they are matching with this. So capital N capital T small u, capital N capital T small u, and it will be small n small t capital U, small n small t capital U. So let's see it. Capital N capital T small u, capital N capital T small u, and here also small n small t capital U, small n small t capital U. So it's matched. So it is 21 plus the DCO, that is 600 to 48, 19.35 centimorgan. So the distance between T and U is 19.5 centimorgan. So N T U, the distance between N and T is 14.5 centimorgan, and the distance between T and U is 19.35 centimorgan. So from this type of sum you can understand the middle gene and the gene and the genetic map and the distance between the genes. Okay, and the formula of the formula to check out the gene distance is number of recombinants by total number of offspring into 100%. In the next class we will understand how to take out the interference, what is interference, what is coefficient of coincidence, and how to take out expected DCO, observed DCO. Okay, I hope you understood it. If you have any queries you can ask me in the comment section. I am giving you one more sum to practice at home. You can take a screenshot of it and you can solve it at home. Okay, this is one sum for your homework and this is another sum for your homework. In next class we will discuss the answer of these two sums and we will move to the next part, that is to interference and coefficient of coincidence. Thank you students for listening, and as I always say, fight for your dreams will fight for you. Thank you.